Serum protein extraction. Processing of serum involves blood collection and separation, sonication of serum, depletion of high abundance proteins, and precipitation of proteins. Carefully withdraw around 4 ml of intravenous blood into a vacutainer tube. Place the tube on ice for an hour immediately after collection to allow the blood to coagulate. Centrifuge the tube for 10 minutes at 2500 rpm to separate the coagulated blood. The blood cells, along with the clotting factors, form the pellet, while the serum containing proteins of interest forms a clear supernatant. Transfer the supernatant containing serum into fresh tubes. These can be stored at -80 degrees Celsius until required for further use. Remove the serum sample from the freezer at least 15 minutes before performing the experiment. Place it on ice and allow it to thaw before use. Then transfer the required amount of serum into a fresh, clean tube. Sonication. Dilute the serum sample 5 times with phosphate buffer of pH 7.4. Vortex the sample to allow complete and uniform mixing. Place the tube on ice and sonicate the serum by providing 6 cycles of 5-second pulses at 20% amplitude with a 59-second gap in between each cycle. Depletion of high abundance proteins. Serum contains several proteins in a wide range of concentrations. Of these, albumin and immunoglobulin G are found to be the most abundant. These proteins may interfere with the gel pattern and experimental analysis while trying to detect other proteins present at lower concentrations. It is therefore preferred to remove these high abundance proteins before electrophoresis. Depletion columns for removal of high abundance proteins are now available commercially. Open the cap of the column, place it in a tube, and centrifuge it after addition of phosphate buffer. Discard the liquid that remains in the collection tube. Then, add the binding buffer provided with the kit to the column and centrifuge the tube. Next, add the serum sample to the column and incubate on ice for 5 minutes. This allows the high abundance proteins to bind to the affinity matrix of the column while the rest of the proteins remain unbound. Again, centrifuge the column and collect the serum that remains at the bottom of the tube for further processing. Protein precipitation. Transfer the serum depleted of high abundance proteins into a fresh tube. To this, add a mixture of trichloroacetic acid and acetone and mix the contents well. The solution gradually becomes turbid due to precipitation of proteins. Place this tube at -20 degrees Celsius for at least 4 hours before centrifugation to enable complete precipitation of proteins. Then remove the tube and centrifuge the contents to allow the proteins to settle down as a pellet. Carefully discard the supernatant and dry the pellet at room temperature. Reconstitute this dried pellet with the rehydration buffer. Vortex the tube to ensure uniform mixing. Store the reconstituted protein solution at -80 degrees Celsius until performing the next step of protein quantification.